Good afternoon. I'm Marc. So my presentation today will be very brief. I'm going to give away right away the take-home messages, and I have 3 today. Number 3, it's all about the males. And if we want a collective answer to red blotch disease management, let's practice some dancing. All right? So now you know everything I'm going to talk about. I can leave the stage and you can enjoy the rest of the afternoon. But more seriously, we know very well that research doesn't operate in a vacuum. It takes outstanding collaborators to carry out research projects. And I'd like to, if the technology works, if I know how to make it work, recognize today Monica and Hannah and the other people in Monica's group for their outstanding collaboration over the past few years. And not only does it take outstanding collaborators, it takes outstanding grad students. For that, I'm grateful, because it takes a village to be successful. I'm grateful to each and every one of you in this auditorium today for having supplied insects, sticky cards, for sharing your little story to me, for confiding in whatever observations you have about what red blotch means to you guys, how does it spread and so forth. Because I always said I'm in an ivory tower, and the more I learn from you guys, the better off I'll be in a position to eventually help you better with your business management. And I'd like also to thank each and every one of you for contributing directly to the CDFA Pierce's disease and glassy wing sharpshooter research program, because you might not know, through your contributions, you're directly shaping up my research and extension program at Cornell. And these are 3 outstanding grad students that I was able to recruit in my program. And everything I know about red blotch biology and ecology, it's because of them. So, red blotch, you heard me say it many times, and I will say it again. Red blotch is not only novel, red blotch does not behave like anything else we are familiar with. Forget about making analogies with Pierce disease. Forget about making analogies with leaf fraud. We have to empty our hard drive and start rewiring everything the way we see, everything the way we think about red blotch. So it requires a whole village to start thinking outside the box. Or as I always say, start thinking outside the bottle. Because if we don't think outside the bottle, we're going to have to repeat mistakes after mistakes. And that's not what we want to do. So today, the planning committee asked me to address how does red blotch spread? And what are some of the factors that influence red blotch spread? And are there nuances between vineyard blocks? And you already the answers to most of these questions. But can we explain why there are so many nuances in red blotch spread? Can the behavior of the only known insect vector so far, which is the 3 Cornell alfalfa hopper, explain some of these nuances? And what are the lessons learned from our understanding of how red blotch spreads? Can some of these lessons be used to develop disease management responses? So, for the sake of time, I'm going to go back to my favorite Cabernet Franc block, a 5-acre block in Rutherford. This block was established in 2008. In 2012, the vineyard manager noticed disease symptoms. 2012, barely anyone knew what red blotch was all about, right? But in 2013, I tested some of these vines and confirmed that red blotch was indeed present in that block. Why that block was of interest to me is, you see close to the riparian area there, the clustering of diseased vines? That called my attention. And naively, I thought, well, the virus is moving in from the riparian area. That explains the clustering. But I was way off, as many times, because the planting material explained the clustering there because we did sequence the virus present in every of these vines in the clustered area, and they were all genetically identical. If an insect vector were to transmit them, you wouldn't see 100% genetic homogeneity. You would see some variability, right? And that was not the case. And the vineyard manager saw the disease symptoms 4 years post-planting. If the cyan were to be the source of the material, of the virus, he would then seen the disease in year 1 or year 2, the first and second leaf. So the rootstock was the issue there. Because when the rootstock is the source of information, of the virus, sorry, it takes 3 to 5 years for the virus to translocate through the graft union, build up in the cyan, and for the cyan to build up to express disease symptoms. So anyway, I wanted to better understand how does red blotch spread. And I was fortunate enough to do it in that block for 10 consecutive years. And obviously, when I talked to the vineyard manager, the winemaker at that state, and I said, you know, take away that clustered area because we never know, it might act as a transmission hotspot. They didn't listen to me. Why would they listen to me, right? Why would someone listen to a French-American hybrid? plus works out of Big Red at Cornell University, right? It's not even here. But I'm grateful to him that he didn't remove it because, again, I could monitor every vine in that block for 10 years. So every white cell you see here is an asymptomatic vine. Any red is a symptomatic vine, so infected. So I'm going to fast forward very quickly, so buckle up, it'll go very fast, 10 years forward, right? And every year, a newly infected vine has a different color, all right? Think about this aggregation there that might potentially serve as a transmission hotspot. 2014, 15, 16, 17, 18, 19, 20, 21, 22, 23, and we stopped there because we ran out of beautiful colors. So, and I didn't say that the bottom graph is the same representation of the vineyard, but across rows, okay? So what really is obvious here is that we always have two distinct epidemics going on in the same vineyard. So one that started with just maybe 20 vines that were aggregated. And boom, it exploded from there. That's the right-hand side. The left-hand side or the west side, it's random distribution. It's salt and pepper type of distribution. So when you think about the increase, on the left-hand side, you have less than a 1% annual increase. Right-hand side, you have less than 6%. So yes, we all have this conviction that red blotch spreads fast. You remember the first talk this morning? Red blotch, leaf roll, the annual rate of increase can be up to 10, 20%. I never seen that with red blotch. Red blotch doesn't spread fast. And I will tell you later why we have the impression that it spreads fast, but it does not. So anyway, yes, when you go for 8% to close to 90%, that's humongous. But it represents only a 6% increase, annual increase. So what we learned that block over 10 years. The planting material was the primary source of the initial inoculum. And that was brought up during the previous conversation. I'm not saying that currently it still is, but we have to be mindful that it has the potential to still remain the primary source of inoculum that is introduced in a new block, particularly the rootstock. And at that time, and I'm not saying that what we learned in that block applies to every block you guys are managing, but the initial aggregation disease vines explained by the infected planting stocks. I'm going to not to read the third bullet point there because I already explained it. What we have learned is, yes, what you guys have talked during previous growers panels. There is virus coming in from the outside. But that's not how red blotch is primarily spreading. Red blotch is spreading within the vineyard. It's the inoculum that is in the vineyard that is getting spread. On top of that, there is a little bit of influx from the outside, but very little. It's the within spread that is explaining the rate of increase we see. There are two factors that influence the rate of spread: the level of disease prevalence in any particular block, and the population density of the 3-cornered alfalfa hopper. These two factors are key to explain fast spread or slower spread, or very little spread, or almost no spread. Because how many times have we seen a fully infected block adjacent to a less infected block, and there is barely any spread here? And sometimes it spreads almost like wildfire. So that's one of the explanations. And yes, we talked about, and previous speakers, who actually, by the way, all qualify as fantastic grape virologists, I was very impressed. Asymptomatic infections are unique to the red blotch situation. I'm not saying that for leaf roll they do not exist, but they do not exist at the magnitude where we find it in red blotch infected vineyards. And I will come back to that in a second. Because again, as mentioned, this is a hidden inoculum that we don't see with our naked eyes. That is very critical in understanding why in some corners of a block you have spread that occurs very fast, and in others it might not occur. Now let's come back to my first takeaway, number 3. And I think that's the most important on that slide. Weather events affect the rate of spread. This is, again, something very unique to red blotch. I'm not aware of any equivalent where there is statistically supported correlation between weather events and disease increase 3 to 4 years later. Let it sink for a second. What I'm trying to say is, what we see visually today is the result of an event that happened 3 or 4 years prior. And I will come back to that, how weather influences the rate of disease spread. Because obviously it doesn't affect the virus, it doesn't affect the vine, it affects the 3-cornered alfalfa hopper. So the other thing that is very puzzling, even in my Cap Franc, we know that the 3-cornered alfalfa hopper flies, right? That was part of our conversation so far. But are there other modes of dispersal? What are we missing? Because how can you explain that if you have an aggregation, it becomes a transmission hotspot if it flies? The transmission hotspot is vine-to-vine transmission, right? It's not something that flies. So are we missing something? Well, how about we look very closely how the 3-cornered alfalfa hopper really moves? It flies, yes, confirmed. But that's not how it preferentially moves. It moves by walking. That's how it preferentially moves, by walking and jumping. So that dispersal movement explains the vine-to-vine. That's why avoid, and yes, Ashton, you, Jake, you said it, avoid any aggregation, because that aggregation will serve as a hotspot that will expand the disease incidence. And yes, I said it's all about males. Damn it. Nothing new, right? Under the sun. Males are the preferred flyers, not females. And males are the primary contributors of spread because they are the primary carriers of the virus. Don't ask me why. That would require another PhD student to understand the difference between the two, but that's a reality. And I will come back to that just in a couple of slides. So everything we know about the three-cornered alfalfa hopper biology was learned 50, 60, 70 years ago from field crops, alfalfa, peanuts, soybeans, in Mississippi and Tennessee, a long time ago. Can we use this information and expect it to equally translate in the behavior of a three-cornered alfalfa hopper in a vineyard ecosystem? Yeah, we know better, right? Obviously not. So we basically hypothesized that the three-cornered alfalfa hopper relies on several plant hosts other than wine grapes, and they use them as feeding, reproduction, and overwintering plants. And the way we did it is, you might know the say that if you tell me what you eat, I will tell you who you are. And it's basically what we did. Thanks to your contributions, hoppers were collected from Napa to Calistoga from 21 to 23. And we tested individual specimens to see what plant material they had ingested and what plant material they had in the gut. The idea is, if we understand the plants they have in the gut, we know which plant species they visited prior to them being caught in a vineyard, okay? And that will give you an idea of a handful of plants they visit before they go to a vineyard, their preferred feeding and reproductive hosts. So our findings were quite interesting, and this was done over 71 vineyard blocks. And roughly over the 2 years, 20% of the specimens carried the virus. There were more in 21 than in 22, as you can see from the slide, and we'll come back to why later. And again, what I illustrated, these damn males. More males carry the virus than females. So what did we find? By far, the most plant that we found in the gut, by far, were esteraceae. Then, second, you have fabaceae, grapes, and many others. So altogether, believe it or not, we found over 2 growing seasons more than 150 plant species in the gut of the three-cornered alpha hoppers caught in vineyards. Over 150. It's amazing, right? So indicating that this hopper is very polyphagous, meaning it eats on almost everything. But the plants we found, mostly in their gut, are not grown in vineyard settings. They are in the surroundings. They are in natural habitats. They are in ditches, in dirt rows, in riparian areas, in woodlands, and these kind of things. Barely grown in vineyards. Barely part of cover crop mixes. So that's just a couple of examples of each of the top plant families that we found. Now coming back to seasonal variations. Again, roughly 150 different plant genera and species listed here. See the big difference in the number we found in 21 versus 22? Do you remember the difference in weather patterns in 21 versus 22? Can you recall? What was 21? Dry, wet? What was 22? Dry, wet? Hot. So 21, if I'm not mistaken, was very dry, right? Look at how few plant species were found in the gut in 21. So if they are in surrounding vineyards, it's dry, it's hot. These plants are dead, right? So where do they go and feed? They come to vineyards. At green, they're irrigated. That's the only food they might have. And you remember, we found more insects that carried the virus in 21. In 22, we had less. It was a wet year. You see how many more plants they have in their gut? They spend, again, the time outside. They don't spend as much time in vineyards when it's wet. They don't need to. They have the preferred food outside vineyard settings. So now I wanted to hone in a little bit. As I said, grapes are one of the preferred feeding hosts. And we know they don't like wine grapes. But as mentioned by the previous panel, they like free-living grapes, right? Vitis californica and hybrids. Let's see what they had in the gut. So here there are 3 types of grapes. Wine grapes, Vitis vinifera, Vitis californica, pure Vitis californica, or hybrids of Vitis californica and wine grapes or Vitis californica in rootstocks. See the difference again in wine grapes between 21 and 22? Spend more time. We found more wine grape DNA in 21, barely any in 22. Again, illustrating that when it's damn dry, they spend more time in vineyards. When it's wet, they spend much, much less time in vineyards. But again, that slide, these 2 pie charts illustrate, again, they don't like wine grapes. They love Vitis californica in hybrids. They love free-living vines. So the panel mentioned, yes, we remove them because some of them are infected with red blotch. Yes, that's a good reason. A better reason is let's get rid of the wine grapes, the free-living grapes. I will come back to removing wine grapes later. But these 2 pie charts illustrate the reason to remove free-living grapes, because they love free-living grapes. And actually, they reproduce on free-living grapes. That's a reason to remove them, okay? So what have we learned? You may recognize the spot where I took this picture, right? So what we learned about red blotch disease ecology, it takes a village to explain what we observe. It has to do with obviously vineyards, adjacent vineyards that might be more red than my vineyard. It takes woodland habitats, riparian habitats, natural habitats, because that's where the 3-corner alpha hopper thrives. And it takes the influence of weather events, particularly rainfall in the spring, more so than in the winter, to explain some of the disease spread dynamics we have observed. So I will not go through this slide, but I do not recommend any action whatsoever against the 3-corner alpha hopper for the reasons that are listed here. It's only a transient visitor in vineyards. It doesn't reside in vineyards. And it's usually visiting primarily in June, July. It's not an efficient vector of red blotch. So I don't recommend taking any action in vineyard settings. So what have we learned, and how can we translate what we have learned in terms of red blotch epidemiology, in terms of virus management? If I don't recommend taking any action against the 3-corner alpha, I do vehemently recommend reducing the virus inoculum. What you guys have been doing so far, kudos to you guys, you're on the right track. Now, it was talked about what is an acceptable threshold cutoff value as to when I should take action. I teamed up with economists many years ago to come up with that kind of theoretical cutoff. And I just put it there as a guideline. Because to me, and we talked about regional approaches, neighboring approaches. To me, red blotch is so different that the best approach should be tailored to each vineyard block. There is no one-size-fits-all, even in a given estate. Each block is different. Each block has a different history. Each block has a different disease prevalence. Each block has a different population density of the hopper that is there. You know best how to take action. It has to be tailored to individual blocks, okay? So 30% is was a study that was done on CAPSOV, whereby if the disease prevalence is higher than 30%, you're better off ripping out the whole block because you're losing your money. And roguing if it's less than 30%. But let's come back to roguing. What are we talking about roguing? Come back to the favorite Cap Franc, right, in Rutherford. And I divided that block just artificially in 5 sections, A, B, C, D, E. And for 3 years, we tested individual vines in these A, B, C, D, E subsections. I will just keep D and E because there I come back to what I told the vineyard manager at that time. Man, remove the entire area. No, I tell him, remove that entire section because, and he agrees. Now finally he agrees. Wow. It took him 10 years to listen to someone. Let's focus on sections B, C, and D. These sections are in front of the epidemic, right? And probably that sounds familiar to you guys. So what is happening there is usually incidence varies between 5 to 20% in A, B, and C. So theoretically acceptable in terms of fruit quality and wine production. But in A, the diseased vines are aggregated. And this takes into account not only the disease vines that are infected, but the asymptomatic vines that are infected. And there we recommend, based on what we have learned about spread, zonal roguing. What is zonal roguing? Zonal roguing is a new concept whereby you eliminate disease vine and some of the neighboring vines, whether they are infected or not, or whether they're diseased or not. In that case, less than 20% disease incidence, but the plants are spatially aggregated. We recommend to remove the red vine, the diseased vine, one on each side within the row, and 2 across 2 rows. So like a cross, okay? So basically we recommend removing 7 vines. In B and C, there's roughly the same amount of disease, 20%, but there is no aggregation. The disease vines are spatially aggregated. There we recommend removing the diseased vine and 8 surrounding vines, basically removing a square, okay? So that's what we have learned. But more importantly, in terms of disease management, and I'm very serious, if we all work on our rain dance and make sure these damn hoppers remain outside of the vineyard settings when it rains, that's the best management option that I can recommend today. I know that's not what you wanted to hear, but let's practice in our rain dance, okay? Thank you.
Marc, we have some questions for you first, if that's all right.
Sure.
Some questions on Zoom are, Marc, do you recommend planting an irrigated trap crop hedgerow of sorts with non-vitus plant species outside of vineyard blocks to prevent them from feeding within the vineyard in the dry season?
No, the short answer is no, because I understand the rationale for implementing such a practice, but it's a very risky one because whatever you might attract to the vineyard might not behave as you anticipate, and suddenly you have an increased population in your vineyard, although that defeats the purpose of the new practice. So I would not recommend it because it's way too risky.
Another question on Zoom is, in the wet years, did Tika feed more on wild grapes because they were well watered in the riparian area in that season?
No, the short answer is no, because in the area there, the river goes dry, so they wouldn't have their feet in water. I think it's just free-living vines are preferred hosts over wine grapes. And I'm not sure to understand why, but we started with chemical ecologists to understand if wild grapes might emit certain chemicals that are attractive to the three-cornered alfalfa hopper versus wine grapes that either lack the capability to produce these molecules or the opposite, produce repellents. So that's a line of research that we're pursuing as we speak at Cornell. With the idea of, let's assume there is a molecule or molecules in wild grapes that act as attractant to the three-cornered alfalfa hopper. If we were able to synthesize chemically these molecules and we were able to spray them in riparian areas, that would be an elegant way to keep the three-cornered alfalfa hopper away from vineyards.
All right, thank you so much, Marc.
Thank you.